Ah oui. 23 participants, oui.
On peut peut-être commencer si vous le souhaitez.
OK.
Okay, so hello everybody. So that's our first Neurobinar of the year, so welcome. And we are going to start with Alicia, who is doing her PhD in the Center for Magnetic Resonance in Biology and Medicine, le CRMBM à la Timone. And she's going to talk about the sequela that we can observe in the brain in a patient who suffered of cerebral malaria. So Alicia, it's your turn.
Okay. Hello, what I'm going to present is...
I know it was not what I got in my little summary. So
I'm going to present about Lisa's
okay.
PhD topic. So
Okay.
I will share my screen.
Yeah,
Can you see my screen properly?
Yeah.
Okay.
So it's
Okay. So hello, good afternoon. I'm Alicia, and I'm going to present actually the PhD project of Lisa Raoul, which is the other girl that will speak today. And she's a fourth-year PhD student at the LNC, and her PhD is called Bodily Self-Consciousness Malleability During Adolescence. So to start, we are going to define, So first, we need to define what is bodily self-consciousness. So in our everyday life, when we're awake, we are aware of the external world, the world outside of us, and we direct our attention, our awareness towards the objects or the person, the people that are in this outside world. But at the same time, in a more implicit way, we're also conscious of ourselves, or of being a subject, of being a body. And this is more a non-intentional consciousness that to most of us is self-evident. And it actually relies on our brain's ability to integrate multiple sensory information, so multisensory information, and it also relies on high cognitive functions. And it's been proven that disturbance in these mechanisms can predispose to certain behavioral disorders like anorexia nervosa. And given that in adolescence, the neural basis of this multisensory process and these high cognitive functions is not yet mature, so the neural bases are not yet mature. Even this, given the case, bodily self-consciousness has not been very much studied in this adolescence period. This is why the goal of her study was to investigate how the bodily self-consciousness adjusts during this adolescence period, which is a period where the body changes a lot. So precisely, she is investigating, or she has investigated, the malleability or the robustness of bodily self-consciousness using virtual reality. So basically what they do is they induce a thing that they call embodiment, thanks to an avatar that they create exactly with the size and the body shape of the subject. And then to test, they test to see how different incoherence conditions are going to affect the embodiment that the subjects feel. So again, to induce the embodiment, they put the virtual reality glasses for the subject. The subject sees the avatar, and they do asynchronous stimulation in which a red ball touches the leg of the avatar at the same time as the researcher touches the leg of the subject. So like this, it's a synchronized feeling, and it helps with develop this feeling of embodiment with this avatar. And then they do incoherence conditions to see how embodiment is affected. So the first way that they introduce incoherence is through a tactile way. So basically, the touching that the subject sees in the avatar and the one that it feels is not at the same time. So this induces this synchrony, and also a visual incoherence. So basically they change the avatar, they change the body mass index, or they change the state of development. So it's more an adult avatar or a child avatar, and then they see how embodiment is affected. And to check the embodiment, they use two measures. Onset time, which is a measure that they do during the simulation. Basically, the subject has to indicate when they feel like the ball is touching their own body. So they see the onset time of dissolution. And then after the condition, they pass a questionnaire to test the ownership feeling. And then the subject receives some questions like, I saw my own body, and they have to rate how much of this was true for them. And they induce the illusion in 70 females aged from 10 to 17, so from early to late adolescence. And the main hypothesis that she had was that bodily self-consciousness is more malleable at the beginning of adolescence, and it becomes more robust towards the end, so with age. And given the two asynchronies that they, the two incoherence, sorry, that they introduced, they expected that the asynchrony effect, the effect that asynchrony had on embodiment was lower at the beginning of adolescence, and it grows towards the end of adolescence, and the same happens for the effect that changing the avatar had. So first, to consider what effect does changing the avatar have. In fact, it had an effect over the ownership feeling, so the questionnaire that they did after the study. And as you can see, the early adolescents, so the younger adolescents, the effect, the difference that the different avatars have over these ownership feelings is not much. So they all have around the same effect, but this changes as the adolescents become older. Okay, so there is a relationship between age and avatar manipulation. In fact, the effect that this manipulation has is more important with age. And they saw that the ownership towards the normal and the adult avatar were the one that was increasing, in fact, with age. So with this, they could conclude that the bodily self-consciousness is less malleable with increasing age, or is more malleable in the younger adolescents. Then they tried to solve the effect of the asynchronous condition. And in here, they saw that what was affected was the onset time, so what they measured during this study. And in fact, here you can see pretty clearly that there is also a relationship between age and asynchrony. In the younger adolescents, the onset time between the synchronous and the asynchronous condition is not much difference, and this difference is much bigger in the older adolescents. So this means, again, that the bodily self-consciousness is less malleable with increasing age, because the asynchrony had more effect on the embodiment in the early, the younger adolescents. And finally, I forgot to mention that they also did some sensory tests to the subjects, and this is because they wanted to see if there were any inter-individual differences that could influence this bodily self-consciousness. And they saw, in fact, that it was interoception that had an effect. So they measured the interoception with a heartbeat counting task. So they told the subject to measure their own heartbeat without touching their body. And then they measured the actual heartbeat, and they got a score of high interoceptive people to low interoceptive people. And they saw that there was a relationship between asynchrony, embodiment, and interoception. In fact, the effect that asynchrony has on embodiment still remains the same as before. So there is higher effect on embodiment with increasing age, but also with increasing interoception. Okay, so the malleability decreases with age. So the bodily self-consciousness is more robust in the older adolescents, and also even more if these adolescents have high interoception or high interoceptive acuity. So in conclusion, she has concluded that there is the bodily self-consciousness is more malleable in early adolescence, and it becomes robust with age. And this is very important because it helps characterize this early adolescence as a period of vulnerability for disorders that involve bodily experiences. And also, she found that interoception may have a role in this vulnerability that she saw. And yes, so this is it for me. Thank you so much for listening. And I think you have any questions, you have to direct them to Lisa, if I'm not wrong.
So thank you very much. I forgot that in fact you were talking about the work of your colleague. It's Alzheimer effect, you know.
It's fine.
So do we accept some question, or because me, I have one. Why do you choose only female in your study? Why not male? And if you add male, do you expect to have some differences?
It's a question. Thanks for your question. It's a question that's often come. Why do we choose only female? So there is different reason, and the first big one is because it's so different between female and male. Like if you look only at the pubertal scale, female are being more height in the pubertal development way before than male. So if we want to study that in male, we have many differences also at the social aspect, because it's not precise here, but we quantify some behavioral aspect of the subject, and on social and psychological aspect, which is also really different in male and female. So we just need way more subject, like double of subject, if we want to study both. And it would be also really interesting to compare between the two, of course, but it would be like another study.
Okay, thank you. So is there some more question, or we continue?
I think we can continue, and then
Okay.
everybody can, ask question after both girls have.
Okay,
spoken.
so now I will not make mistakes. So we are going to listen to Lisa, who is going to talk about the results of Alicia, about the sequela of the neurological sequela of patients with cerebral malaria. And as you understood, Lisa is coming from a Laboratoire de Neurosciences Cognitives, and I give you la parole now.
Okay. So. Can you see my screen correctly?
Yes.
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 Okay.
Yeah.
So I'm gonna present Alicia's project, and Alicia will try to characterize the brain cycles that are related to experimental cerebral malaria. So first of all, what is cerebral malaria? So malaria is nowadays, sorry, I just have a problem. Okay. So malaria is nowadays the worst parasitic disease in the world, and its most lethal complication is called cerebral malaria, which happen during the infection by what is called Plasmodium falciparum. It's lead to this in 15–20% of the case, which are mostly children under 5 in sub-Saharan Africa. And there is currently no treatment for cerebral malaria, and the vaccine that was authorized really recently, only in 2021, is only efficient in 30% of the cases. Cerebral malaria cause sequestration of erythrocytes in capillaries of the brain, blood-brain barrier damage, and severe brain edema. This can be seen here in the MRI of the child here in the slide. That you can see here, a child with cerebral malaria and a child without cerebral malaria. And as of clinical feature, it causes seizure that eventually may lead to coma and death. A very important thing concerning cerebral malaria is that it causes neurological sequelae that can be developed right away, persist in time, and even appear years later after the disease has long passed. So they come in many different forms, as behavioral disorder, cognitive impairment, motor, sensory, memory, and language deficit, epilepsy, ADHD, etc. The most important point is that there is very few studies that have report clinical and preclinical reports of the long-term neurological outcome of cerebral malaria. So this sequelae, which contribute to the human, social, and economic burden of malaria, are largely unstudied. The aim of Alicia's project is therefore to use in vivo MRI and MRS to characterize the neurological sequelae of cerebral malaria at the anatomical, metabolic, vascular, and functional level on the long term. She also wants to look, as a second goal, she wants to look into the difference between males and females in terms of how they are affected by the disease and the sequelae they experience after recovery. So to do that, for this aim, she used the model of cerebral malaria, with survival in male and female, called C5T7 black 6 mice, infected with Plasmodium berghei onca, and treated here with chloroquine at disease peak. So after she injects the Plasmodium berghei, she tried to see when the malaria happened, and then she started the treatment. The animals are explored with magnetic resonance imaging and magnetic resonance spectroscopy technique before and after the disease, and then once a month during 6 months here after recovery. And at the end, she do behavioral test and brain extraction, but I will come back on the next slide. So as per the specific technique she used, she will acquire anatomical imaging here to see the general state of the brain structure, brain perfusion maps here, okay, brain perfusion maps here, angiography of the brain vessels, diffusion tensor image to study the water diffusion in the tissue and with that the structural connectome, and resting state functional imaging to study the state of the functional connectome. She will also perform localized proton magnetic resonance spectroscopy and phosphorus magnetic resonance spectroscopy to study the neurometabolism in the hypothalamus, hippocampus, brainstem, and cerebellum. Additionally, she will include behavioral test of the survivor animal and a post-mortem study of the brain extracted with NMR to further acquire information on the metabolism of the brain after the disease has long passed. The expected results she have for this project are based on previous results from the group and the literature on the sequelae of cerebral malaria. And the expected results include, first, a persistent microvascular dysfunction in the brain of cerebral malaria survivor. Secondly, she expects an alteration in the metabolism of the brain because of tissue damage and functional anomalies. Then she also expects a long-term alteration in the structural and/or the functional connectome that persists even after the disease has disappeared and the parasite cleared from the blood for a long time. Finally, she also expects to see a difference since the study is done in the two sexes and based on the report of the human disease. She expects to see difference in the way male and female are affected by the disease and experience the sequelae. And here are some preliminary results that Alicia has so far. So first, she saw that female and male present difference in how they are affected by the disease in terms of clinical signs, as for example, they take longer to recover the normal value. They also see difference in the survival with a higher death rate here in male if you compare to female, and location of the brain lesion, which are different here in male than female, like you can see here and here. Also, she found that the brain lesion that she observed in the mice that have undergone cerebral malaria and survived it are maintained even after 6 months after the clearance of the parasites that you can see here. These are observed in the olfactory bulbs of the ventricles, among other regions, and also here. So, and also she found finally that survivorma presents in the behavioral test higher anxiety levels and lower cognition memory than the control 6 months after recovery. And I thank you for listening. And yeah, that's it. And I think now you can ask questions to Alicia if you have some.
So ask somebody some question. Me, I can't see. So maybe Gabrielle.
There is a question from Anne to Lisa. Do you think that the fact that the effect of asynchrony is lower in younger may be due to a difference in multisensory integration processing with age?
Okay, so I answer that, or we take Alicia's question, or I answer that also?
It's a question for
okay.
For you.
Yeah, for sure. It can be that, but also of course, yes, tourism, it can be it can have an effect. The ability of multisensory integration can have an effect on that. But as we test, also we did not present, but we test multisensory in tactile and also in proprioceptive integration, and there is literature that it's growing, it's developed differently during adolescence. Maybe we can look at that and see the difference.
So
I have a question for Alicia. You showed, Lisa showed that males and females have different types of lesions as observed by MRI. And my question is how reproducible and consistent are the lesions between males, different males, or between different females? Is it really reproducible? Is it always the same, or it's quite variable?
It's quite variable. It's true that so far I have still a lot to analyze because I'm still in like the experimental phase. Some of the problem with the lesions is that they're very hard to, so we're still finding like a way to a classification. So a way to say how we measure, like, for example, there's a lot of microhemorrhages in the olfactory bulbs. So we want to find a way to like count the points that we see. There's like a high point density. But so far I cannot tell you. I know I understand your question, but so far I cannot confirm if it's really consistent. I have seen different lesions, but I don't know yet if this will really like yeah.
Is it only me?
Sorry?
I lost you for 10 seconds. Maybe it's from my side.
So yes, so I did
the last 5 seconds I missed. So it's
yes, so I did see different lesions, but so far I have not found a way to properly quantify it or classify the images. So I cannot tell you if this will maintain.
Thanks.
Itself.
Me, I will have maybe a curiosity question. So, do you inject the virus, the malaria? Where do you inject? In the blood, in the brain, or?
I inject intraperitoneal, so in the
okay. So quite similar to when a mosquito is.
Yes. So in fact, what I inject is not the parasite. I inject already the red blood cells infected by the parasite. So I am like being the mosquito in this case.
So now I understand better. Thanks.
You're welcome.
There is some question in the Q&R section. So one from Pascal. Thank you both for the presentations. Question to Alicia. Which kind of behavioral tests will be used in your project? Do you expect behavioral differences between males and females?
Yes, so I use the elevated plasmase, so to test anxiety, and also object recognition, novel object recognitions to test the memory. And so far, I have seen differences between the groups, but I have not seen difference between the sexes. But because I have not yet tested many males, so far the test I have passed them in some females, but not in many males. Although I do believe, just by observing the mice, that they will have even higher anxiety levels. Just by like observation, but this is not an official value yet.
Thank you. Two other questions for you, Alicia.
Okay.
One from Anne Cavounou-Diaz. What do you expect from diffusion images? Alteration in some specific tracts?
So I will not be doing tractography. So I am doing diffusion weighted. So probably I will have information on medium diffusion, MD, medial diffusion, I think, fractional anisotropy, and all this. So what I expect is increased diffusion in some regions where there was lesions during the disease time. So I'm also going to see this, like compare the lesions that I have seen during the disease, like the peak of the disease, with the diffusion values in these areas, which I expect that there will be more diffusion, which is normally indicative, or less fractional anisotropy. But I am not, I think, no, I'm not going to do tractography. So probably I will not be able to see the specific tracts that are affected.
Thank you. Another one for you, Alicia, from Adriana Perles. Alicia, is the cerebral blood flow as seen on the MR angiographies and or cerebral perfusion maps restored to normal after the peak of the CM disease?
So what I see in the angiographies, so basically all the blood vessels, I only see visually, so I really don't have a way to quantify it. And visually, it does go back to normal. So this is like the big vessels. But then the perfusion, I see the microvasculature, and I have seen so far, still I have to like analyze so much more data, but I have seen so far that the perfusion decreases, and it maintains decreased at least the 6 months that I have done the follow-up. It doesn't really go up to the normal values that I took before I induced the disease. So this I don't see in the angiography in the big vessels, but in the microvasculature, at the microvasculature level. So far, it doesn't go back.
Okay, thank you. If there is some other question later. Alicia and Lisa can answer afterwards by mail or whatever. I think it's time now to give the words to Antoine. Agnes, can you do a small presentation of our next speaker? Thank you.
I will try. So Antoine did his PhD some years ago in Paris under the supervision of Pierre-Marie Ledo, and he was working on the molecular control of the adult neurogenesis in the olfactory bulb in the mice and rats. And then after this PhD, he went to Harvard. And so yesterday night, we took a little bit of time to summarize what he did in Harvard. And if I understood well, in fact, he tested the hypothesis of the potential possibility of adult neurogenesis to be used as a cellular therapy in Parkinson's disease. But apparently he stayed quite a short time in Harvard. And so I don't know where is the hypothesis now. And so after Harvard, he came back to France and joined the Harold Kremer lab at the IBDML. He stayed quite a long time there. In 2012, he entered CNRS. And in Harold Kremer lab, he was working on the transcriptional and post-transcriptional mechanisms regulating neurostem cell, and he focused on dopaminergic neurons. So maybe something about Parkinson's disease was still in his head. Then in 2017, already 6 years ago, he joined INMED, and he was where he was working on cortical development in health and in neurodevelopment disorders. And recently, he published paper about he developed a single cell RNAseq approaches to interrogate abnormal development and function in rodent models of neurodevelopment disorder. So I can see Antoine moving his head. So I don't know if it's happy with what I say, but now you have la parole, and you will explain much more than me what you do.
Thank you very much, Agnes, for the
welcome
introduction. And it was mostly true what you said, mostly right, except that my work on single cell RNAseq is not published yet.
Ah. Well, it will be.
Okay, so now thank you also, Gabriel, for the invitation. And so let me talk to you about our current work on the molecular control of cerebral cortex development that has been done in great part by excellent PhD student Rémi Mathieu, who is now a doctor, and in collaboration with Ludovic Tollet, who has his own lab in the University of Lausanne. He's an expert in single cell RNAseq analysis. So we are interested indeed in cortical cerebral cortex development, the molecular mechanisms of it. And so the cortex is a very complex structure which is composed of a large diversity of cell types, neuronal and non-neuronal, that develop in a very finely orchestrated manner, such that ultimately each cell type adopts a stereotypic laminar position in the 6-layered cortex, as well as a very specific synaptic connectivity with other cells. So while the final position of each cell type and the connectivity between synaptic connectivity between different neuronal types are very well understood, the molecular mechanisms that are at play are basically not known for the great majority. And even the alteration of the connectivity between a single neuronal type and another one can be at the origin of a neurodevelopmental disorder. So the finely orchestrated microcircuit organization is really essential for the function of the cortex. And so we saw that the ligand and receptors mediated interactions between different cell types might be important for the positioning and the connectivity in this structure. because it's the case in many other tissues and organs, and it has to be in a way. And there were some data suggesting that it could be the case, that ligand and receptor could be important, including our own data. For example, in this paper from last year, we found that the disruption of the transcription factor NeuroD2 causes a neurodevelopmental syndrome with autistic features via cell autonomous defects in actually cortical glutamatergic neurons in humans. And we used the knockout mouse to investigate the physiopathology of this syndrome. And we also use conditional knockout mouse in which NeuroD2 was deleted specifically in glutamatergic neurons of the cortex. And the main phenotype we observed was a subtle alteration of the layers, of the 6 layers of the cortex, with basically the layer 5 was expanded towards the surface of the brain at the expense of layer 4. And so it's a switch, superficial switch of the neurons towards the surface. But surprisingly, and it's not published, what Remy found later on was that some GABAergic neurons, inhibitory neurons, like the parvalbumin subpopulation, which is the higher in number, were also switched in position towards the superficie, as is evident from these pictures, I think, and is quantified here. This indicates that the position of parvalbumin neurons, at least, is non-cell autonomously regulated via cellular interactions with glutamatergic cells, probably via ligand-receptor interactions. And this is reminiscent of 2 other important studies that are shown here. And before describing them, let me first tell you a bit more or remind you about the main glutamatergic cell types in the cerebral cortex in terms of axonal projection specificity. There are 3 main types, which are color-coded here. First, you have the intratelencephalic neurons, which are blue, and that express SATB2, and they constitute 100% of layer 2 and 3, and only a minor fraction of layer 5 and 6. And they are intratelencephalic, meaning that they connect the other hemisphere. Second, you have the pyramidal tract neurons in red that are only in layer 5 and express the transcription factor phase F2. And they connect brainstem nuclei and the spinal cord. And third, you have the corticothalamic neurons, which are only in layer 6 and connect the thalamus. And every one of these projection neurons plays an essential role in the function of the cortex again. Here, the team of Paola Arlota, they knocked out the phase F2 pyramidal tract PT marker, and they found that this didn't kill the neurons, but it switched their identity. The layer 5 PT neurons became layer 2-3 IT-like. So they start to connect, for example, the other hemisphere instead of the spinal cord, and they change in molecular and electrophysiological ways. And this was accompanied by a non-cell autonomous change in the positioning and connectivity of some GABAergic neurons, again, non-cell autonomous. And this last, more recent paper from the McBain team, in this one, what they did is to delete the IT neuron marker SATB2 specifically in all glutamatergic cortical neurons, hippocampal too. And what they found here, similarly, is that these neurons don't die, but they switch their identity and they get a mixed IT identity, which is a bit more like the PT-like identities. They start to project in part towards the brainstem and spinal cord. And this indirectly changed, again, both positioning and synaptic connectivity of some GABA neurons. So these 3 studies, to our mind, indicated that glutamatergic neurons control positioning of GABA neuron types, probably via ligand and receptor molecules, right? So this slide is to show you that the GABA neuron types have very restricted laminar positioning, and this is quite recent because for long it was thought that it wasn't the case, that the GABA neurons were broadly dispersed in the cortical sheath. And this was because we didn't look in great detail in the fine subtypes of GABA neurons. We thought for a long time that there were like mostly 5 types of GABA neurons, which are depicted here. You know, the parvalbumin and somatostatin, which arise from the medial ganglionic eminence, and these 3 types that emerge from the caudal ganglionic eminence, which are in the ventral brain, and they colonize the cortex later on, after the glutamatergic neurons. But now, with the advent of single-cell genomics, and in particular patch-seq, we can know a bit much more. So the patch-seq is a technique by which you can patch neurons, like to measure their electrophysiology. But in the meantime, you can also aspirate their cytoplasm and therefore determine their molecular signature and determine their transcriptome. And this way, by the molecular signatures of the different cells, for example, if you focus on the P-valve family or subclass, parvalbumin, they could, the Allen Institute researchers, they could find by the patch-seq that there were different types of parvalbumin neurons, a lot of them. And second, thanks to the patch-seq, because they knew where the neurons they patch were, they could determine that they were restricted to quite specific laminar positions corresponding to 1 or 2 layer maximum. So there is very restricted laminar positions, but the molecular mechanisms at play, not clear at all. And for the connectivity, synaptic connectivity, here we draw an example which is the non-exhaustive of the connectivity in layer 2-3. So in green, you have the glutamatergic neuron, layer 2-3 IT, intratelencephalic. This is the only one in this layer. And the other cells are most of the GABA neurons that are connecting it. And you can see that depending on the subtype, it connects it at different subcellular levels. For example, the parvalbumin CHC corresponding to chandelier cell connects the axon, while the parvalbumin basket cell, another subtype of parvalbumin, connects the soma. Another type of basket cell, which is a CCK, many labs study it because it's very important to connect also the soma. And then you have the Martinotti cell, which is a subtype of somatostatin, a large family, connecting the apical tuft. And finally, for example, I can just stress this one, which is because it's quite studied and quite important for cortical computation. You have the vaso-intestinal peptide B-tufted cell, which connects almost exclusively other GABA neurons, but not glutamatergic neurons. So it's a disinhibitory neuron. So you can see that there's very specific connectivity patterns which need to exist in order to have a normal function of the cortex. But the molecular mechanism during development that address axons to specific dendrites in the targets are not known. So this is our question. What is the molecular code? The long-term question, it's not answered yet. By which glutamatergic neuron types control the positioning and connectivity of specific GABA neuron types. And if we broaden a bit the question on the longer term, we would like to try to understand better what would be the molecular code for the establishment of a stereotypic cortical organization. And so there will be two parts. First, I will show you how we generated a bioinformatic atlas in order to predict or infer ligand receptor interactions between the cell types over cortical development. And second, how can we use this atlas? Now, for the first part, I will first show you how we managed to track down each post-mitotic neuron type over cortical development at the transcriptional level. We used the single-cell RNAseq approach, and so this is the experiment, the detailed experimental paradigm. So what we did is to perform single-cell RNAseq from the somatosensory cortex. We focus on this region on a time period spanning P0 to P30, because it was first lacking from literature mostly. Second, because it corresponds to the lamination of the GABA neurons that arrive in the cortex after the glutamatergic ones. And third, also because it corresponds to the window of establishment of synaptogenesis of all the neurons. And this is what we did, this study here. This is a Sankey plot, so meaning that the thickness of each line corresponds to the number of cells that we got, okay? And so from our study, we focused on the somatosensory primary and secondary cortex. And we performed single cell and single nucleus RNAseq. For some time points, we used single nucleus because we didn't have some cell types with single cell RNAseq. And then we used 10x genomics drop-seq approach to perform the single cell RNAseq. So for those who don't know, this is a nanofluidic platform with 3 main entries. In the first entry, you put individual cells. In the second entry, you put barcoded bits, so which have unique barcode, DNA barcode for each bit. and in the third entry you put oil. And in the end, the scheme is a bit not right, but you end up with oil droplets, and in each oil droplet you have one cell and one DNA barcode that can allow to recognize it in the end. And you lyse the cells, and you can get the mRNAs and measure the sequence, the transcriptome of every cell with very high throughput, 10,000 cells in one experiment. So we did that. And so we decided to integrate data sets that were published by recent papers that are listed here in order to be able to follow the different types of neurons from E11 to adult. So there were data sets from earlier and other from later time point. So this one is the data set 2001 from the Allen Institute that the molecular biologists, cellular and molecular biologists know. They did a lot of cells. And this is very comprehensive ones, and they annotated, they gave a cell type name for all cell types. And then we also added the DBELA study from last year, in which they did many time points every day from E11 to P4 in the somatosensory cortex. You can see they did exactly the same region. And we added other regions that basically correspond to the ganglionic eminences, because we saw that this would allow us to track the GABA neurons even before they reach the cortex, when they are in their zone of origin in the ventral brain. And so in the 17 time points, and the big challenge was to be able to assign a cellular identity for each neuron that we could track over time. The same, to use the same nomenclature or taxonomy for every data set. And it was not that easy. And we used as a reference the Yao paper, which is shown here. So what they did is they performed single cell sequencing for all the cortical and hippocampal regions. You can see in these little schemes here. But we subsetted, we took only from this somatosensory cortex, and they have 80,000 cells, so it's quite a lot. The way they identify the cell was using a hierarchical approach, which allows to have a high confidence in the cell type annotation. So I will go a bit quick on that. So this is a dendrogram basically, and each vertical line to correspond to a cell type, and two lines that are close correspond to cell types that are close transcriptionally. And so at the highest, the less resolutive level, you have the class that was assigned. So non-neuronal, glutamatergic, or GABAergic. Okay, and then if you go for each class to the subclass level, so the higher resolution, you cluster inside the class. For example, for the non-neuronal class, you would have the astro, oligo, microglia, endothelial, smooth muscle cells. For the GABA, you would have the parvalbumin, somatostatin, and the CGE-derived subclasses. And then you can go another level of clustering to go to the supertype level, which is correspond to these circles here. So it's becoming quite resolutive, and the identities are shown here. And those supertypes, it's a good level of resolution because you can kind of make the correspondence between this level and the biologically meaningful cell types that were identified thanks to the patch-seq, in which they had the electrophysiology, the morphology, and the transcriptome of the cells. So they can determine really the cell types that make sense at the biological level. And the cluster level is very granular, very high granularity, but we don't really know what these cell types mean. So we wanted to reach kind of the super type level for our data sets. So to make a long story short, we decided to keep only post-mitotic neurons, and we didn't take into account the non-neuronal cells afterwards. And for each independent data set, we were able to determine the two glutamatergic and GABAergic class. And the trick afterwards was to be able to compare the gene expression between cells of the different ages. And for that, we had to use what we call integration. So it's a bioinformatic technique in order to get rid of the batch effect or technical bias, which is huge because you can imagine we are dealing with data sets done by different people in different labs from different regions with different RNAseq methods and a platform for library preparation and even sequencing machines. So we want to look only at biological variation and not at technical bias. So this integration was done, and in the end, we end up with 160,000 neurons, and that can be represented in a UMAP here. So it's a UMAP is like TSNE, it's a dimensionality reduction methods, which basically you represent in 2D all the cells, and basically each dot is one cell, and two cells that are close to each other correspond to cells that are transcriptomes that are very similar. And then you can color code this representation using whatever codes you want. Here we coded by class. You can see that the two classes are well separated, so it makes sense. You can then color code by age. And so you have embryonic in red, neonatal in green, adult in purple. And it's quite logical and it makes sense that you can see that the very young neurons, post-mitotic neurons, are always in the center of the UMAP. And then when you go from the center to the periphery, you go through age, basically. And it's kind of logical. And we were able, by many hard work of computational methods, to go from the class level to a family level that corresponds to the subclass, and then to from the family level to the supertype level, by doing this hierarchical approach. And from the supertype, we made the correspondence with these two seminal papers that use PatchSeq to get the morpho-electrotranscriptomic information about cells, allowing them to give them some biologically meaningful cell type names. And this is what we did. In the end, we get 27 cell type names, which are shown here from these families, 11 glutamatergic types, comprising 5 IT cell types. I'm sorry, it's a lot of nomenclature, but this kind of work is all about that. We have to describe the most of this project is to try to really describe in great detail how the cortex is organized. And then we will be able to develop hypotheses. But that's why there is a lot of nomenclature to learn here. So you have the intratelencephalic neurons that correspond to the IT family. You have the extratelencephalic family comprising the pyramidal tract and the corticothalamic. And then you have more other cell types that are more rare, like the chiral retius that are transitory, and these two cell types that are quite rare. And you have 16 GABAergic neuron types, which are the most well-known by experts in the field, especially people at INMED. For example, if you focus on the parvalbumin family, you have 4 cell types. You have the chandelier and the basket. And among the basket cells that connect that are perisomatic, you have the ones from the different layers. So now, for downstream analysis, we will be able to look at all these cell types, at their expression of ligands and receptors. And you can distribute all these cell types in the UMAP. And it's quite neat to see that the glutamatergic neurons are on the left, while the GABA are on the right. And the NGE derived are below, and the CGE derived are above. So it kind of makes sense. And just by doing the correspondence between our own 160,000 neurons and this PatchSeq data, and also spatial transcriptomic data sets, which is the future of single-cell transcriptomics, because now we have spatial information of the cells in the tissue, we were able to predict very precisely the laminar position of each one of our 27 cell types. Now, I will go very quickly in this part where I will talk about spatiotemporal transcriptional dynamics. And so what we can do here is to embed all cells in a 2D graph from one family. For example, if we take the IT cells from the glutamatergic class, in this 2D graph, and in this 2D graph, we have in X, it represents a normalized and continuous time, while the Y represents pseudo space, which is a normalized and continuous space, let's say. And you can see each dot is one cell. And you can see that there are 5 cell types among this family, and there are a lot of time points. And actually, there is a transcriptional gradient of variation in both time and space. But the point here is that you can now plot the expression of any gene of interest in this kind of 2D representation. And therefore, you can get this spatio-temporal expression map in which, for example, CUX1, it's a known gene, for the protein corresponding to the gene is known to be expressed in the superficial layers in IT neurons. And this is what we confirm. Here we can see that it's only in superficial layers, and it starts to be expressed at E16 until the end. And so you can do this kind of spatiotemporal expression maps for any type of family. Complexin 3 is a subplate marker. It's a transitory marker, so we can confirm that it's not expressed late in life. Somatostatin is more in the middle, in the superficie. It's not in the deep layers, but it's already present in early time points. And parvalbumin is known to be expressed very late. It's not expressed in the embryo, even in neonates. So it all makes sense. And now we go to the important part, inferring ligand receptor interactions. And so the simple way to do that is just to cross our gene expression data for all our cell types with a ligand receptor database containing 6,000 or a bit more ligand receptor pairs. And this way we can predict basically ligand receptor pairs that could mediate positioning and connectivity patterns between cell types of interest. And since we have 27 cell types, we have 27 multiplied by 27, which makes almost 900 cell type pair possible, and we can infer interaction between any of these cell type pairs. And from this, we generated an atlas with which we can infer the interactions. I think I will have to skip this because it's too long. And so in the end, we have a shiny app, and it will be a website available to the community called Single Cell Ligand Receptor Somato Development. In which you can, before to look at ligand receptor interaction, you can visualize clustering results. You can look at all the cells, and you can color code them based on whatever you want. You can look at only subclasses, supertypes, cell types. You can look at the spatiotemporal expression of any gene, not only ligands and receptors. So I think it can be interesting for many people, for any cell type family over time and space. And then coming back to the ligands and receptors, you can visualize the number of predicted interactions at a given age. So here I plotted the 27 cell types as a source, ligand expressing source, and the 27 cell types as a receptor expressing source. And I ordered the cells based on the cell types, on the class, the GABA, the 16 GABA types and the 11 glutamatergic types, and also I ordered based on a superficie depth, normalized soma depth. And then you can see that, for example, there are many interactions that are predicted, and the chiral ratios that are known to be at this time to interact a lot with glutamatergic cells indeed do interact with a lot. There are lots of ligand receptors that are predicted between chiral ratios and all the glutamatergic cells, but also with many of the GABAergic cells, which is less studied to my knowledge. And then you can go a little bit deeper and query or interrogate the atlas for specific ligand receptor pairs and do dot plots where you have intracellular and intercellular scores that are proportional to the expression of ligand and receptors and also the downstream target genes of the receptor. So you can really go deep. Now, in the last 5 minutes, sorry, I'm a bit long, I will show you how we started to use this tool. The first, we can use it to validate previously demonstrated interactions, and we took the example of neuroregulin 3 ERB4. So this paper and many others have shown that this ligand receptor pair is very important for glutamatergic synapse formation onto GABAergic neurons in the cortex. Neuroregulin 3 being expressed in glutamatergic presynaptic terminals, while ERB4 in the dendrites of GABA neurons. And so, but what can our atlas tell us about this? Can it confirm this prediction, these functional experiments? Well, yes. If you look first at the spatial temporal expression maps, you can see that neuroregulin 3 indeed is expressed by the different glutamatergic types, and actually it's expressed only late in all layers from layer 2, 3 to 6, but not early, only late. You can see high expression in IT and ET neurons, intrateloencephalic, stratoencephalic, during synaptogenesis, so it makes sense. And ERB4 is confined to GABA neurons. It's not expressed by GLUT neurons. And among these GABA neurons, it's almost restricted to somatostatin and parvalbumin during synaptogenesis, late. And in somatostatin, it's in all layers. In parvalbumin, it seems to be more in the superficial. So it's already something new here. It has to be, of course, validated by experiments, but it's an indication. Now, if we query the atlas for cell pairs that are predicted to interact via this neuroglin 3 RB4 interaction, we get this. If we put neuroglin 3, anyway, it's expressed in GLUT neurons of upper and deep layers, and RB4, it's expressed in GABA neurons in red. For upper and deep layers. And we can see that there are predicted interactions between most glutamatergic and most GABAergic neurons, basically. It's a lot of dark squares. Only these mice2 and long projecting somatostatin cells, they are not predicted to interact through this pair with the glutamatergic cells, but it's not surprising because there are very different types of inhibitory neurons compared to the other ones. Yeah. And if you dig a bit deeper, you can interrogate the expression, and you can see that there's a lot of interaction predicted again between GLUT and GABA neurons. And actually that this interaction is increased between P1 and P8. You can see that the size of the circles is higher, there are more circles, and the color of the circles is stronger. This is the intracellular score reflecting the downstream signaling genes of the receptor of RB4. And so it is very coherent with the role of RB4 in synaptogenesis, because P8 is the boost of synaptogenesis. So conclusion, our atlas validates what's known about this pair. Now, can we use the atlas to complete knowledge about non-interactions? And so we were interested by this paper, in which they found that some of the somatostatin GABA neurons need to release cereblin 4 to form synapses onto GABA neurons, glutamatergic neurons, sorry. But they didn't know which type of somatostatin, because there are 4 types, nor what was the receptor. And so we thought maybe we can know more with our atlas. And we first looked at the expression of the ligand cereblin 4. We found there's non-significant expression in all families except the somatostatin family. And actually inside the somatostatin family, we found it's expressed only, it seems to be expressed only in layer 2-3 fan-Martin T cells, but not in the 3 other cell types. But this is something new that needs to be verified. And if we interrogate for ligand receptor interactions, the only one that is predicted is between this somatostatin fan-Martin T population and most GABA neurons. Most of them, except two exceptions, and the chiral ratios, so it's normal because they disappear. And if we look at the higher resolution, you can see first that there are 4 receptors for cereblin 4 that are known from the literature, and that among these 4, only 2 are predicted to mediate interactions in this case. And among these 2, GLUG1, which is an AMPA subunit receptor, and neogenin1, this one seems to be the best candidate because it's strongly increased at P8, which is a time point where the synapses are being formed. The conclusion for this part, and then I'm almost done, is that with our atlas, we are able to predict answers for the 2 questions of these papers that can be tested now, that are being tested by experiments. And now the Graal for us is to find from scratch new ligand receptor pairs that would be mediating positioning or connectivity patterns in cortical microcircuits. And so here, I'm not coming back because I removed them. But you can see if we focus on cadherins, the family of cadherin genes, actually there are many members that have differential expression inside given families. Some of them are only early, some of them are intermediate, some of them are only superficial, some of them are only deep, like cadherin 13, etc. Suggesting that they may be important for the patterning of the different positioning, connectivity things. And just to, as you know, cadherins allow mostly homophilic interactions, so 2 cells that express the same cadherin tend to interact. And this has been hypothesized to control synapse specificity, especially due to expression to the expression in different layers or cortical areas. But there is no really proof of that. It's just an hypothesis. So we focused on cadherin 13, which is restricted in glutamatergic IT neurons in the deep layers and late during synaptogenesis, but not so much during positioning lamination. And in GABAergic neurons, cadherin-13 is also late. It's also expressed during synaptogenesis more than during lamination, but it's not restricted to the deep layers, at least for somatostatin, where you can see it's in all layers. For parvalbumin, it's enriched in deep layers. But anyway, if you look at the red rectangle, from this, our hypothesis was that cadherin-13 might be important for the connectivity pattern, synaptic connectivity between the IT neurons and their partners in these deep layers, which are the Martinotti or basket cells, parvalbumin. And the second reason why we wanted cadherin-13 is that because mutations in this gene are associated with neurodevelopmental disorders. If we interrogate the atlas for predicted interactions, there are only predicted interactions in the deep layer glutamatergic neurons, not in the upper layers, 0. And the interactions are predicted with only few GABA neuron type, which are parvalbumin layer 4-5 basket cell, with all the GLUT actually, and with the somatostatin, Martinotti, and non-Martinotti cell. And this I don't have time. But to make a long story short, what we found by expression and by prediction is that cadherin-13 is not expressed in the chandelier, for example, in the deep layers, but only by the basket among the parvalbumin family, and it's expressed in the glutamatergic and in the Martinotti cell. It's never really expressed in the superficial layers, at least for the glutamatergic. But our question was, what if we knock down cadherin-13 in the deep layer glutamatergic neurons, which is the purpose of the lightening of the purple pointillé? Do we get a reduced innervation by the basket cells? We focused on the basket cells and not on the Martinotti cell because we have a good marker of the connectivity of the synapses made by basket cells. And so what we did with Anusha and Leah was to electroporate at E13 an shRNA against cadherin-13 combined with GFP in order to reduce cadherin-13 in deep layer glutamatergic cells. And then we sacrificed the animals after birth at P28 to allow lamination and, more importantly, connectivity to occur. And finally, we looked at synaptotagmin 2 positive puncta, which correspond to the basket cells presynaptic bouton onto the soma of the electroporated cells, in order to look if cadherin-13 reduction would reduce the perisomatic inhibition. And indeed, you can see here, maybe on this image, that you have a reduction in the number of presynaptic puncta if you reduce cadherin-13, but the size of this puncta is not altered, which is this graph. And this confirms our hypothesis. And this was also confirmed by a poster at the FANS this summer from the RICO lab, who is expert in the field. And now we are collaborating with them on that, actually, in which they found by another approach that in the deep layer 5 neurons, glutamatergic neurons, if you reduce cadherin-13, indeed, you reduce the perisomatic innervation as measured by synaptotagmin 2. And so this is to us a proof of principle that we can find new candidate molecules that regulate at least connectivity. And now we are focusing on some other cadherins that might regulate this time more the positioning of the cells. But this is ongoing work. And so I'm sorry I was a bit long. I learned only yesterday that it was 20 minutes. I don't know why I thought it was 40 minutes. So I tried to reduce, but anyway, most of the work was done by Rémi. And I thank you for your attention.
Yes, thank you very much, Antoine. So I tried to make some. Okay, so if I can start with two questions. So first question, so you reported that you have a diminution of PV terminals around the soma of pyramidal cells, but do you have the same in your neuro D3, D2 KO as you show at the beginning of your talk? You know, where you show that the PV cells were differently distributed, and have you checked the perisomatic innervation of pyramidal cell in this kind of model? That's the first question. And the second question, okay, you check the number of the terminals, you say that the size is not different. And do you have any idea of what happens to the postsynaptic side on the GABA-A receptors? If you have a difference and things like that.
The first question for the NeuroD2 knockout, we didn't look, but we don't have specific reasons to think that cadherin 13 is reduced in the deep layer of the NeuroD2 knockout mouse. What we have in the NeuroD2 knockout is that there is actually an acceleration of the radial migration of the neurons that over-migrate. And as a consequence, the PV neurons that are supposed to follow a specific types of glutamatergic partners, they follow them. That's what that's my hypothesis, and that's why they over-migrate too. But I don't know if it's only cadherin 13 that mediates that. And what we think is that cadherin 13 is important for perisomatic innervation in deep layers, but that maybe other cadherins are important for the same perisomatic inhibition in superficial layers.
Okay.
And so we don't think it's a change of genes too much in the NeuroD2 that mediates this. We are not sure that there will be a modification in the perisomatic inhibition, because we think that actually the inhibitory neurons, they follow their partners. And so
I understand.
Correct.
But they follow, maybe they can connect differently, and okay.
It would be interesting to do. It's just I don't have enough hands. And the second question
was the effect on the postsynaptic side. You know, do you have any idea of what can happen with the GABA-A or maybe GABA-B receptors?
Yeah, you're right. It's a quite superficial analysis for now, and we should do a co-labeling with maybe Gefferin or GABA-A receptor.
Gefferin will be a very good idea to see.
We did that, but we had some trouble, and we don't have yet quantification, but it's something that we should actually pursue, yes. And we should do the electrophysiology also, of course.
I'm neuroanatomist, you know, so it's another world.
Yeah.
So are there some other questions in the audience, maybe?
There is another question, but what I propose is I've copied the question and make an email between Antoine and Ibrahim.
Yeah, because it's late
so that you can answer directly to Ibrahim, if it's okay for you, because a lot of meetings are
yeah.
So thank you very much.
Everybody.
That was so interesting, the three of you. Thank you for doing that for us.
I'm sorry I didn't respect the time limits. I didn't read the mails in detail. It's my fault.
You're passionate. That's what is important. Thank you very much.
Bye.
Okay. Cheers.
Thank you. Bye.
Thank you, everybody.
Thank you.